pandemic sucked, but we also managed to develop an mRNA vaccine in a record-breaking 11 months. What makes mRNA so powerful? Let's use SARS-CoV-2 as a case study.
SARS-CoV-2 uses its spike protein, a key, to bind to the cell receptor, a doorway into the cell. Then it inserts its genetic material and replicates, some viruses leaving the cell to infect other ones. To combat this, our immune system generates a cell-mediated response, destroying infected cells, and an antibody-mediated response, targeting viruses outside the cell.
However, instead of all of this happening during the attack, wouldn't it be better to teach our immune systems how to react beforehand? That is the role of vaccines.
Traditional virus vaccines contain weak, killed, or partial parts of a virus, and when injected into our bodies, develop immunity. However, this requires the growth of the virus in a laboratory for consistency, known as cell culture production, a very long, complex, and expensive process.
Well, what if scientists got rid of the entire cell culture process, no longer injecting the actual virus into our bodies, but instead tricking our bodies into making parts of the virus itself? Enters messenger RNA.
First, some background. In a normal cell, messenger RNA or mRNA plays a vital role in translation between DNA and proteins. This process happens in two steps: transcription and translation. Transcription begins in the nucleus of a cell, where DNA is translated into a strand of mRNA bases. This mRNA strand then travels from the nucleus to the ribosome, a protein-making factory. This is where translation occurs. Transfer RNA reads the mRNA strand's bases in groups of 3, known as a codon, and then retrieves the bases' corresponding amino acids, which together form a protein.
But what does any of this have to do with a vaccine? Well, mRNA can code for any protein. So what if scientists make a vaccine with an mRNA strand? What if that mRNA strand codes for spike protein from SARS-CoV-2? Once injected, this mRNA vaccine would command our cells to produce spike protein, which our body would recognize and learn how to fight against. And then in the future, in a real infection, the spike protein would be blocked. And that right there is the breakthrough, because not only would this mRNA vaccine replace cell culture production, but it also most closely simulates a real COVID-19 infection where the cell itself produces the virus. As a result, our immune system will generate not only the antibody-mediated response that a traditional vaccine induces, but also a cell-mediated response, leaving us even better equipped to fight a real infection.
This breakthrough might save us in future pandemics, and is even being used to develop treatments against cancers like melanoma. And hey, that only took 3 minutes to explain. If only a vaccine could be developed that fast.